Welcome back to Anatomy Physiology 1, Chapter 4, Part 2, where we're talking about DNA, and here is a reminder of transcription. So this is where RNA is copied from DNA. Once the messenger RNA has been transcribed, then you remove the exons and introns. You recombine the exons and they exit the nucleus. The introns remain in the nucleus. And here's a little cartoon I found that I thought was funny. So here's your ribosome. Here's your messenger RNA, and it's going through the ribosome, through the little groove there. I call it the sombrero hat. And in this one, it's just making arginine, 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 which you abbreviate ARG, ARG. So this is a pirate ribosome. On one end of the transfer RNA is an anticodon that matches with the codon of the messenger RNA. On the other end of the transfer RNA is the amino acid that's going to be making the protein. And you're going to have to join the amino acids. So here is a cartoon showing you the dehydration synthesis. So you're going to take a hydrogen off of this one, an OH off of this one, and water will leave. And now you have these two amino acids attached by dehydration synthesis. And we call that particular bond a peptide bond. Here's a nice little summary of all the things we talked about. The DNA is the boss. It's a double helix. It opens up, makes the messenger RNA. Messenger RNA needs ribosomal RNA to slide through. Transfer RNA is bringing in the amino acids. The RNA, if you can't make proteins, get out of the kitchen. So you have three kinds of RNA and DNA involved in protein synthesis. Just as the messenger RNA is not complete once it is transcribed from the DNA, you have to alter it. You're going to cut out some pieces and splice it in different ways. You're going to do the same thing to the proteins that you have made by doing translation at the ribosome. So you have to have the correct primary structure, which is the order of the amino acids, and the secondary structure, if you remember, was the beta pleated sheet or the alpha helix, so it can coil up or it can look like a Lay's potato chip. And then there are chaperone proteins which help push the different strands of amino acids into the proper shape, the three-dimensional or third-dimensional folding. And if something happens and your your protein starts to denature, what you can do is you can create heat shock proteins that will help fold your damaged proteins back if they're not irreversibly denatured. Now, when you fry an egg and you have that white runny yolk and then it turns into that kind of rubbery fried egg, you're not going to have any kind of a heat shock protein that's going to turn that back into a liquefied egg white again. This is a nice summary of what we learned in last chapter, and it kind of goes and ties into this chapter. So we learned about the nucleus, and we learned how the rough endoplasmic reticulum is closely associated with both the nucleus and the cell membrane. And here they're showing where you're making proteins using the ribosomes that are attached. And they're going to go through the lumen, they're going to go through the tunnel, the pathway within the endoplasmic reticulum, and then it's going to bud off and send it over here to the Golgi apparatus, and the Golgi apparatus is going to modify, add things, take things off, and once it is correctly assembled, it's going to break it off into these vesicles, and it can send them over to the edge of the cell membrane, and it can do exocytosis to release whatever that particular protein is out into the interstitial fluid. So to summarize, the rough ER has ribosomes that are making proteins, most of which can be sent out of the cell, can be packaged by the Golgi apparatus and sent out of the cell. But you also have a lot of free ribosomes in the cytoplasm, and they're mostly used for making proteins that you need within the cell. So they're used in-house instead of exported. Not all of the post-transitional modifications are made by the Golgi apparatus. Some of them happen within the endoplasmic reticulum itself. For example, you can add sugars, you can add carbohydrates to it. You can stabilize a protein with disulfide bridges. So if there is a cysteine and another cysteine, then they can join and share sulfurs, which will hold the protein in a three-dimensional shape. I mentioned that not all of the genes are turned on at the same time. So some are permanently turned off, or we hope they're permanently turned off, inside of cells, so that a cell that's busy doing muscle type things is not also busy trying to do bone kinds of things too. But another thing is you may need the genes, for example, in your breast to make milk, but only if you have a baby. So you don't want those genes, even though they do have something to do with being a breast cell, you don't want them turned on at an inappropriate time. So we have hormones that can turn off and on. So there's a whole new science that they just came out the last decade or so called epigenetics. And it's out of the scope of this class. But if you want to do some extra reading, go read Epigenetics, EPI genetics. I'm going to spend a minute or two on this particular slide because there's a couple of things that I think are important. One of them is, this is a nice overview of some of the things we've been talking about. There are receptors on mammary cells, in the breast cells, and human prolactin can bind to those receptors. So they're on the outside of the cell, but they can trigger a protein on the inside of the cell membrane. This protein moves to the nucleus. It can bind to the DNA near the gene, and once it's bound, then the RNA polymerase knows where to go and where to start unwinding the DNA so that you can transcribe it into messenger RNA for casein, which is one of the milk proteins. And then this messenger RNA moves to the cytoplasm, finds a ribosome on the rough endoplasmic reticulum, and you're going to make the casein, and you're going to secrete it because you secrete milk into the ducts in the breast. So the cells make this and then put it out in the ducts, and then the ducts lead out to the nipple where the baby can get the milk. The reason I also wanted to go through this, the second reason, is there is a thing called lactose intolerance where you can't break down milk sugar. So some people say, well, I'm allergic to milk. If you're truly allergic to milk, then it would be something like the casein, the proteins that are in it. But lactose intolerance, those people can't break down the milk sugar. So we've looked at how genes code for proteins, but we synthesize a lot of things besides just proteins. So the glycogen that we form, which is a bunch of glucoses that are stuck together by dehydration synthesis, which if it was a plant, we call it starch, but since it's us, we call it glycogen. The fats, some of the fats we have to eat, some of the fats we can synthesize within our body. We take cholesterol and we turn it into steroids estrogen, testosterone, progesterone, hydrocortisone. And then we take the fatty acids and we attach them to half of a sugar molecule, which is a glycerol, and we make triglycerides, or in the case of phospholipids, it's 2 fatty acids and 1 phosphate group. So you have the fatty acids that are hydrophobic, and you have the phosphate end, which is hydrophilic. And that's how you end up with your phospholipid bilayer. You have 2 layers with the lipids in the middle and the phosphates on the outside. So you don't have a direct gene coding for them, but you do have genes that turn on, and then they make enzymes that can then cause these things to be made. So their synthesis is called indirect genetic control. The next thing we're going to talk about is DNA replication in the cell cycle. So most cells are just hanging out, doing cell things. They're reading the chromatin. They open it up, read the genes, do protein synthesis. So they do all the normal maintenance things that a cell does. But perhaps a skin cell is knocked off and you need to replace it with another skin cell. Now the cell is going to have to undergo DNA replication, and then you're going to have to go through the cell cycle of mitosis. We spoke earlier about the 4 different bases, the A, the C, T, and G, that are on DNA, and the A, C, U, G that are on RNA. If you omit one of these bases, if you put in the wrong base, then you're going to alter a gene. Now remember that only about 2% of your DNA is coding, and the other 98% is doing other things. So hopefully if you're going to have a mix-up when you're doubling your DNA, when you're replicating your DNA, it'll happen in the non-coding region. So in order to double your DNA or replicate your DNA, first thing you do is you have to unwind it off of its histones. And then you're going to unzip it so that the 2 strands, the complementary strands, are separated. And then you're going to build new strands using each of the 2 other strands as a template. And then once you've done that, then you're going to have to wrap the new DNA around some histones again. So you're going to repackage it. The name of the enzyme that unzips the DNA is called DNA helicase. That kind of makes sense because it's a helix. You talk about the DNA helix, and ACE, anytime you see ASE, you know you have an enzyme. So you know that this enzyme is doing something to the DNA helix. In this case, it's unzipping part of it so that you can copy both sides of the DNA. If you take a biochemistry course, then they will definitely tell you all about the replication fork and how you're reading one strand of DNA in one direction and one strand of DNA in the other direction. And they'll name the DNA polymerase, and they'll talk about the other enzymes that are involved in this. What I'd like for you to know is that you're going to pull the DNA apart. You're going to have the 2 strands separated. The A's and C's and T's and G's will come in a complementary fashion, and you're going to make 2 new strands. Now, you don't make it all in one chunk. You make it in pieces, and then you have an enzyme called ligase that puts the pieces together. So here's a cartoon to help you remember what ligase does. It makes ends meet. It joins pieces of DNA together. For us to replicate all of the DNA that's in one cell will take us anywhere from 6 to 8 hours to make duplicate copies of every single strand of DNA. Bacteria are so much faster. they can do the whole process in less than 20 minutes. But then they only have one chromosome, and we have 46 chromosomes. At the same time you're doubling your DNA, you're doing DNA replication, you're also making histones, because once you've made all of that DNA, you've got to have histones to wrap your DNA around, because we're going to have to package it back into chromatin. So here's a kind of a cartoon summary of the whole thing. So here's your DNA, and you're going to have a DNA helicase, which is going to pull the two strands apart. And then you have room for the DNA polymerase to come in, and it's going to be reading and making a double strand. And then going in the opposite direction on the other strand, you also have an enzyme that is replicating the DNA, making a second copy. So you're going to start out with one double strand, and you're going to end up with two identical double strands if everything goes well. Every time your DNA replicates, you're going to have about one error every 1 billion bases. And you think, well, that's not very much. But then you have to think how many billions of bases you have that make up your 46 chromosomes. So luckily, there are enzymes, DNA polymerase is one of them, that will fix places where it can find errors in replication. Besides mistakes that are made when you're making an extra copy of your DNA, there are other things that can mess up your DNA. For example, radiation, X-rays, viruses can get in and alter your DNA. If you're exposed to certain chemicals, they call them mutagenic chemicals. If your DNA is damaged so badly that your cell cannot fix it, it can kill the cell, it can make the cell become cancerous, or it can cause genetic defects that you can pass on to future generations. And one of the things that worries me a little bit, because one of the things that college students tend to do once they're out from under their parents' fingers is try out recreational drugs. And you should look at the mutagenic properties of things like LSD. Not something you want to be experimenting with if you want to have children. For the non-recreational drugs, you can call your local pharmacy and ask them about certain medications, and they'll tell you whether or not it's likely to interfere with cells that are dividing. There are some medicines you can take that will definitely with the dividing cell, and especially the ladies out there who are having babies, you should try to limit as much as possible any medications that you take. But if you have to take a medication, by all means tell your pharmacist that you're pregnant, and they can look up and see what the chances are that that particular medicine that you take would mutate the DNA for your baby. And we call those terratigens. I never know whether to show you the picture first or the words first, but anyway, here are the words. We're going to learn that the cell cycle has interphase, which is made up of G1, S, and G2, and then mitosis, which is made up of prophase, metaphase, interphase, telophase, and not cytokinesis. Again, this is an error on this slide. Cytokinesis is not part of mitosis. So there should be three steps: interphase, mitosis, and then cytokinesis is its own thing. So I'll bring that up again in a minute because it's going to be really important that you know that. This is as good a time as any for me to tell you about mnemonics, which are memory devices that will help you remember things. You do need to know the phases of mitosis. And the easiest way that I have found is to remember ipmat. Ipmat, I-P-M-A-T. If you can remember the word ipmat, then the phases of mitosis will fall into place for you. An example that I give students, I usually pick a student at random and I point to them and say, okay, name the 5 Great Lakes. And almost no student can do it. They just kind of look at you like, what? And then if I tell them, well, the first letter of the 5 Great Lakes spells the word homes, H-O-M-E-S, then they're like, oh, Huron, Ontario, Michigan, and so on. So a memory device will help you remember. Most of you learned Roy G. Biv for the colors of the rainbow. So you've been introduced to mnemonics before. But if you can remember ipmat, I-P-M-A-T, then that'll help you a lot. So this is a cartoon picture of the cell cycle. And you start out in the first growth phase. So we call it G1. But what they really need to have right here is, this was left off of the picture, so I just went in and I added it. Most cells will exit right here and go to what we call G0. And in that, the cell is not dividing. It's just doing what it needs to do until it grows old and dies. So G0 is a cell that is not going anywhere. It's just doing normal cell functions and not dividing. But if you need the cell to divide, then you're going to go this way. And instead of being in G0, you're going to go into G1, or the first gap phase. This is where the cell grows up, and it does all the things it needs to do to get ready to double the DNA. So that's the G1 phase. Then you get to the synthesis phase, and that's where you pull the DNA apart, the two strands, and you make a copy of each one, so you double your DNA. It's called DNA replication. And then once you've doubled your DNA, remember you're also making histones, then you're going to go over to this phase, which is the second growth phase or second gap phase, and you're going to grow up, double the other stuff that you need. You're going to have extra mitochondria. You need some extra endoplasmic reticulum because you're going to split the cell in half. So you need both halves to have a copy of every bit of DNA, some mitochondria, all of the organelles that we talked about. Once you get to this phase, so you've gone through G1, S, G2, now you're actually ready to go into mitosis itself. So all of this, G1, S, and G2, is called interphase. It used to be called the resting phase, which is such a misnomer because the cell is very, 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 very busy at this point in time. But it is between phases, so the DNA is still in the form of chromatin until you reach this phase. And now you're going to coil the chromatin up so tightly you can actually see the DNA. You can actually see it. So we call this prophase, the very first phase where you can actually see the chromosomes forming under a light microscope. And at this same point in time, you're going to have to dissolve the nuclear envelope. Because remember, you want half the DNA to go one way and half to go the other way, and it cannot go out through the nuclear pores. So you're going to have to dissolve the cell, nuclear envelope. All right, the next phase that you're going to do is you're going to line the chromosomes up through the middle of the cell. So they're going to be attached at the kinetochore to microtubules that will pull the chromosomes apart. So where you are lining up in the middle is the easiest of all the phases because they are literally lined up across the middle. So metaphase is in the middle. And once you start pulling the chromosomes one direction, one the other direction, then you're in anaphase. And once they start pulling in, kind of almost looks like a peanut, then you're in telophase. It's the final phase. And then, after you've done that, the cell itself, the cell membrane, has to split. And that's called cytokinesis. Now, if you make it through all of this, G1, S, G2, and all of this, you get to telophase, and you do not complete cytokinesis, what you're going to end up with is one cell that has two nuclei. And that won't work. So what you've done is you've made a cancerous cell. So you're going to have to destroy that cell. So cytokinesis is extremely important. I haven't noticed students of this generation making up poems, but that was one of the things that we used to do to learn things when I was growing up. One that sticks with me is, "Little Fanny drank a drink, but she shall drink no more, for what she thought was H2O was H2SO4. So H2SO4 is sulfuric acid. If you try drinking it, it will literally eat your throat out. So here's one that somebody made up. They must be from my generation. It says, "A chromosome shaking with dread, to her dear sister chromatid said, though it's breaking my heart, will soon be torn apart by a strong microtubule thread." So they didn't go back and spell check it. Here's a brief summary of what I've just said, that the first gap phase, or first growth phase, as some people call it, is from the time the cell is born until you get through the DNA replication. The cell is going to do normal task, get ready to be a big cell. Once it's got all the materials it needs for DNA replication, then it can go into the synthesis phase. Otherwise, it's going to stay in G0. That's an option. It can stay in G0 and not divide, or it can go ahead and get ready and then go and double the DNA. Once you've doubled your DNA, you're pretty much committed. And then you go into the second phase. Any enzymes that you're going to be needing, any errors that you made when you replicated your DNA, you need to be able to fix those. So you've got a little time there, the 6 to 8 hours, to do that. Some cells are more complicated than others. Some of them take longer to divide, so that's why it's anywhere from 6 to 8 hours to replicate the DNA. On average, it takes about a day for the whole process of interphase and mitosis and then cytokinesis. The end result of mitosis is 2 identical cells, each having 46 chromosomes. Why would you do mitosis? Well, you started out as a sperm and an egg, so you were 1 cell big. And now you're about 50 trillion cells, give or take a few trillion. Any organs, any tissues, you have to grow them. Again, you started out as 1 cell. You got to grow your heart, your liver, your spleen, all of those organs. If a cell dies, you have to replace it. So we've talked about losing skin cells, and we've played around with cells in the laboratory. If you damage tissue, you hit your finger with a hammer. If you get stuck by a thorn, if you get into poison ivy, you got to fix the tissue that you damaged. So you're going to be doing mitosis to replace or grow And here's a cartoon to help you remember that word, mitosis. What did the cell say when his sister stepped on his foot? Mitosis. Here is another cartoon that I thought was kind of funny. Are you happy with your current cell provider? If not, if there's something wrong with your cell, then your body will tell that cell to kill itself. And that process is apoptosis. Here's a cell that is in prophase. And here's two things that let me know that it's in prophase. The number one thing is I can see a nuclear envelope. It's kind of sketchy. Here it's kind of broken up, but I can still see most of the nuclear envelope. I can also see chromosomes. So this is taken through a light microscope, the ones that you use in laboratory, and this is what you see. So this is prophase. The DNA is condensing, the chromatin is condensing, and you're actually able now to see chromosomes themselves. This is a nice picture because it shows you some of the phases. So different cells are doing different things. This cell may be in G0, or it may be doing interphase and getting ready to double its DNA so that it can do mitosis. But this one, I can't see the DNA. I can't see the chromosomes, and I can see the nuclear envelope. So this cell is in interphase. This cell right here is in anaphase, and this one is in telophase. Here's a really nice picture of telophase. You can see that it's trying to turn into two cells. And you can see the cell membrane trying to come in here and separate these out. So it's trying to do cytokinesis. But at this point in time, this is telophase. And the way I can tell it's telophase is there's the chromosomes right there, but there is no nuclear envelope, and it's a set. There's a mirror image across from it. So that's the thing that gives it away. If you just saw this half up here, you can say, well, I don't know, it might be prophase. But when you see this, where they're across from each other, you know that that's telophase, and it's about to do cytokinesis right through there, and then you'll have two different cells. This is a nice picture because it shows you all the phases happening all over the place. So you can kind of look around and say, okay, this one is in interphase because I don't see any chromosomes, and I do see the nuclear envelope. So this one's in interphase. If you look at this one, you go, okay, this is telophase, and the way I know is because you can see two cells right here that are getting ready to do cytokinesis and split. So you have two identical cells right there. Here's another one that's in telophase. It's odd, usually you don't see so many in telophase, but somebody got a really good picture because this is also in telophase. And this has undergone cytokinesis right there. And as you see, the nuclear envelope has come back, and the DNA has unraveled. So now it's chromatin again. So here are two daughter cells right there, identical to each other, that have gone back to either G0 or G1 of interphase. This one is mitosis. You can see the chromosomes lined up across the middle, metaphase of mitosis, because you can see them lined up across the middle. So metaphase in the middle. And usually that's the one that you see the most of. So this is kind of an unusual slide. Anaphase, they're starting to pull to either side of the cell. So there's no nuclear envelope. Nuclear envelope's gone. The DNA is spread out across the whole cell right there. So you know that this is anaphase, and they're being pulled. You can't see the microtubules, but they're pulling. And eventually, it'll pull it to either end like this. Sometimes students tell me they have trouble figuring out whether they're looking at interphase or prophase. So here's a clear interphase right here. There's the nuclear envelope. So it's unmistakable, and you do not see any chromosomes in there. You do see the nucleolus, but you don't see the chromosomes. So this is an interphase, and this is in prophase. So I can clearly see the chromosomes forming. They're still in the center of the cell. If you look, you can still see a little bit of the cell membrane, but it's breaking up. So if you look at the two of them side by side, there's no way you should be able to get interphase and prophase mixed up. Take time in the laboratory to look at some dividing cells under the microscope and see if you can find each of the phases. So here's telophase. Here's half of the DNA pulled down to one end of the cell, and here's half pulled to the other end. So this is telophase. Here, this has divided right there, and you've done cytokinesis, or you're doing cytokinesis. This one is telophase because you do not have the division between them. This one, I do have the division, so these are actually daughter cells. It's finished with telophase, actually. This one is in prophase. I can tell because I can see the nuclear envelope, but I can also see chromosomes. So that's got to be prophase. Another picture of anaphase. Here go the chromosomes towards the ends. And once they get to the ends, then they're in telophase. If you were to look at this, these would all be in interphase. This one's in prophase. This one's in anaphase, because there go the chromosomes, half one way, half the other way. They haven't made it to the ends yet, so they're not in telophase. And here's another slide. There's your interphase. These are all prophase. Here's metaphase. They're lining up in the middle. This is anaphase. So they've let go. There were two copies that were held together by the centromere. And the centromere lets go. One copy goes one way, one copy goes the other way. And so that's what you're seeing here. And you can actually see the microtubules pulling. So these are real pictures, and it's kind of neat. And so prophase, metaphase, anaphase, and there's telophase. And here is cytokinesis, right there, where it's going to split into two cells. From the time the DNA condenses from chromatin into chromosomes, we're going to call each of the daughter strands of DNA, we're going to call them, or sister, we're going to call them chromatids, because they're held together at the centromere. So it's kind of like somebody's velcroed them together. As soon as you line up in the middle, an enzyme is going to come and it's going to separate the centromere. And because they're attached to the microtubules, it's going to pull the chromosomes one direction, one the other. As soon as you get to the end of telophase, you're going to see that the chromosomes are going to start turning back to chromatin, and you're going to see the nucleus reappear. So technically, you can't call a chromosome unless it's in telophase. Before that is either a chromatid because it's held together with its identical copy, or it's unraveled and it's chromatin. So why they like to emphasize that, I don't know, but I'm telling you just because you'll probably see it. I had to throw this in because one of the things that any teachers had to go through, especially if they've taught middle school or high school, is kids giggling when you say certain words. So it's kind of refreshing when I'm teaching anatomy that I can say the word penis or testicle and the kids don't start giggling. But here are cells, and their teacher is teaching them about cell division, mitosis, and they're like, ha, 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 ha. The teacher's like, oh, just grow up. So a couple of important things that you need to know about regulation of cell division. So you can't divide unless you've got enough nutrients and organelles so that both of the cells have something to use. Otherwise, the cell that doesn't have any organelles is going to die. And that actually does happen to certain cells that we'll talk about later. You have to have growth factors that will signal the cell to go through these various stages. And there's a whole series of growth factors that you can learn if you take a biochemistry class. If a cell dies, it will open up a space for the other cell to divide. Otherwise, cells have what we call contact inhibition. So contact inhibition is really important because if a cell is touching another cell, then the signal goes out, there's already a cell there, do not divide. But if injury, you skinned your knee or something like that, and you remove the cells, then there's nothing there touching the cells, and so they'll divide and they'll fill in the gap. So that's how we heal from, getting hurt. If you have a cancer cell, they lose contact inhibition. They do not care if they're touching another cell. So they will divide anyway, and now there's not room enough for all of the cells. So they start pushing the nearby cells. And because they still don't have contact inhibition, they will divide again. So some textbooks say that cancer is rapidly dividing cells. Well, no, they're not rapidly dividing. They still have to go through the process. They have to double the DNA. They have to do all of that stuff. So they're still not going to divide any faster than, 6 to 8 hours for the replication of the DNA and the 24 hours to do the whole process. But what they should be saying is they will divide every chance that they get. So because they've lost contact inhibition. And so you actually end up with a hard, dense tumor. And it's going to send out messages to your body to grow blood vessels in, to nourish it, because you've got this actively dividing cells and they need nutrients, so they're going to need blood vessels. So one of the things they do for chemotherapy is they stop you from growing new blood vessels, and they're hoping to starve the tumor. So it'll run out of nutrients and the cells will start trying to divide and they'll end up dying instead. But loss of contact inhibition is one of the hallmarks of a cancer cell. I got a kick out of this because I'm having students texting me and asking me questions, and that's good. If you're stumped, feel free to text me and ask me a question. And also, there's tutors available. But anyway, I thought this was funny. So here's somebody that texts their teacher and says, okay, can you explain the cell division very quickly? And it's like, yes, here's a cell. The cell gets bigger. The cell pinches in half. And then you have two cells. And the kid says, thanks. There are some chemicals that you're going to need if you're going to leave G0 and go into G1 phase. and cyclins would be one of the things that you need. You normally don't make these, but if you are going to have your cell divide, some point in G1, you're going to make cyclins. And then the cyclins are going to turn on kinases. And the kinases, if you remember, the function of a kinase is to add phosphate to things like proteins. So now you're going to phosphorylate proteins, and now you're getting ready to tell the cell to double the DNA. So this cyclin and the cyclin-dependent kinases are important in whether or not a cell is able to divide. So it's a point that we can control the cell division. If things are working correctly, you can stop and keep a cell in G0 phase so that it doesn't go on to G1. If you're going through and it said, okay, it's all right, go on, double your DNA, go ahead, start into mitosis, then at the point that you get into metaphase, there is a place where you can just stop it and say, okay, we're not going any further, we're not going to divide the DNA, we're not going to split, we're not going to make two cells. So that is another checkpoint where the cell can stop and say, there's something wrong with the DNA, it's not working out. And then there's another phase, a third phase, which allows the sister chromatids to separate. So there are checkpoints as you go along through the cell cycle to keep you from making cells that you should not. For example, cancer cells. I'm going to stop here, and the third and final segment of this chapter will be over genetics.